Hello friends and welcome to CEC live lectures. In the ongoing lecture series on extraction of natural products, in the previous two lectures I have discussed about the basics of extraction procedure, wherein we discussed what are the basic characters of a good extraction procedure. We also discussed about the characters of an ideal solvent that has to be used for an extraction procedure. What are the steps that are involved during the extraction of constituents that are present in the plant matrix? That is how the solvent first diffuse inside the particles or the drug particles, then it solubilizes the constituents and then it diffuses out, and with the help of convection the solubilized material enter into the bulk solvent. We also discuss about certain factors that affect the extraction procedures like particle size, volume of the solvent, drug to solvent ratio, and so on and so forth. We also discuss certain common terms that are used during the extraction, that is mark, that is the material that is left behind, or the drug matrix that is left behind after the extraction is complete. Then menstruum, that is the solvent that is used for the extraction of the drugs, and micellar, that is the solvent which contains the solubilized constituents in it. So friends, in continuation with the lecture series, today I am going to start with the conventional techniques that are used for extraction. And these conventional techniques include maceration, which is sometimes also known as soaking, then digestion, infusion, decoction, reflux, percolation, and soxylate. Now, why we call them as conventional extraction techniques? Because these techniques have been used for extracting drugs since ages, and these techniques are not only used to extract the herbal drugs, but in general they are also used to extract the components, whether from the plant tissue or the animal tissue. So coming on to the first technique, that is maceration, or also known as soaking. So as the name soaking indicates, here we just dip the drug into the desired solvent for a specified period of time. We take a coarsely powdered drug because the particle size, as we discussed, the factors that affect the extraction efficiency, particle size is an important factor. So we will move on to this particle size factor later on. So first I will just briefly give you the procedure of maceration. So we just take the coarsely powdered drug and soak it into the solvent. Now the question arises, what should be the volume of the solvent? So the volume of the solvent, normally we take 1:10 or 1:20 in ratio of 1:20, but by and large, if we look into the overall procedure, then the level of the solvent should be enough that it should cover the uppermost layer of the drug. Because if the level of the solvent is below the drug layer, then the drug layer which is above the solvent will not be extracted properly. So the solvent volume should be enough that it should cover the upper layer of the drug, and it should entirely cover the drug layer. Then this mixture is allowed to stand for specified period of time, which is generally 7 days, but minimum it should allow to stand for 3 days. So once the specified period is over, then we filter the extract, or we strain or we decant the solvent from that mixture, and the mark, which is the material that is left behind, is pressed. The pressing is further mixed with the filtered extract. Now in between, what we do, we can increase the efficiency of the maceration process by shaking or by providing gradual agitation during the standby period. That is, during the 3-day period or the 7-day period, we can give intermittent shaking or agitation to the mixture, which helps in improving the mass transfer during the extraction. As we discussed in my previous lecture, that there is a stagnant film of the solvent that is formed around the drug particles, and for a very short period of time that layer enters into equilibrium phase where no mass transfers occur. So this agitation or shaking of the mixture does not allow that stagnant film to enter into equilibration, which further improves the mass transfer, and as it increases the overall extraction efficiency of the process. So once we we are done with the extraction, we press the mark, we mix the pressing with the extract, then the extract is again filtered to remove any kind of drug particles in it, and finally the extract is concentrated under vacuum or any other suitable technique that has been specified in the method. Now we know that the drugs can be of two categories depending upon the presence or absence of the cellular components, and when the cellular components are present, we call that drug as an organized drug, and where the cellular components are absent, or we call it as unorganized drug. Or examples, if I give you, of unorganized drug is nothing but the gums or exudates which we obtain from the plant. Resins are there, whereas organized drugs, they largely contain the leafy part or the stem part or any other part of the plant material. So what is the difference between the extraction procedure when we are extracting an organized drug by maceration and an unorganized drug by maceration? So in case of organized drug, we extract the drug with the whole volume of the menstruum. Say for example, if you are extracting 1 kilogram of drug, you are taking 10 liters of the solvent to extract that drug. So whole volume means you will put 10 liters of the solvent into the drug and allow it to stand for the specified period of time with intermittent shaking in between. Whereas in case of unorganized drug, we don't use the entire volume, that is 10 liter volume of the solvent, whereas we only use 4/5 of the total volume, which will be in our case will be 8 liters if we are using 10 liter of the solvent. So in case of unorganized drug, to extract 1 kg of unorganized drug, we will first treat that drug with 8 liters of the solvent. Now once the specified time period is over, what we do is we strain off the liquid and press the mark in case of organized drugs. So we press the mark, we mix those pressings into the main extract, and this is the procedure which we follow while extracting the organized drug. And in the end, we do not adjust the volume of the total extract. Say for example, we are extracting 1 kg of drug with 10 liters of solvent, and once the extraction is complete, we have mixed the pressing in the extract, and the volume which we obtained, say it was 9 liters, because 1 liter was absorbed by the drug particles. So we are not going to make or adjust the volume to 10 liters by adding 1 liter of the solvent, because this will basically dilute your extract. It will decrease the strength of the extract. Whereas in case of unorganized drug, which does not contain any cellular mass, so what will happen after the specified time is over, that material will get converted into a sticky or a gummy residue which does not require any pressing. And mind you, we have already used only the 8 liter of the solvent and 2 liter is still left behind. So once the extraction time is over, we just decant the liquid or we filter the liquid. We do not press the mark in case of unorganized drug. And once the solvent is decanted or filtered, the mark is washed with the remaining volume of the solvent, that is the 2 liter of the solvent which has been left initially, that is used to wash that particular mark or the unorganized drug residue that is left behind. And that washings are again mixed in the previous extract to make up the volume. So the big difference is, in case of organized drug, we press the mark once the extraction is complete and we do not adjust the volume of the solvent to the initial volume. Whereas in case of unorganized drug, we do not press the mark and we adjust the volume to the final initial volume by adding more solvent. Because in case of unorganized drug, there is a very less chance of the solvent being absorbed by the plant material, because that is an unorganized material and it does not contain any cellular tissues or the cells which can absorb the solvent. So therefore, volume adjustment will not make any effect on the overall strength of the extract that is obtained at the end. Now we move on to the modifications of the maceration process. As we have discussed, that maceration is simply soaking the drug material into the solvent for specified period of time, which is normally 7 days or minimum 3 days. Now with intermittent shaking in between or agitation which we provide to the mixture. Now we can further increase the extraction efficiency of maceration, because during the tenure of 3 days or 7 days, the mass transfer rate gradually decreases with the passage of time, because the solvent will move towards the equilibrium phase. So in order to overcome this problem, we can go for the repeated maceration, which does not allow the solvent to enter into the equilibrium phase. And repeated maceration can be double maceration or a triple maceration. The overall time period remains the same, that is 7 days or minimum 3 days, but we can divide those 3 days into 2 macerations. Say again, we will take an example of 1 kilogram of drug. So we will extract that drug with 10 liters of the solvent, and the total extraction duration which we decide is 3 days. So for first 1.5 days, in case of double maceration, once the time period of 1.5 days is complete, we will decant the solvent and we will add fresh 10 liters of the solvent into the drug material, and then again we will allow that mixture to stand for another 1.5 day. So total duration is 3 days, and once the 3-day duration is complete, we mix those two extracts and we concentrate it under vacuum. Now when we shift or when we change the solvent, we add the fresh solvent to the drug material, the equilibrium does not attain, and there is a huge concentration gradient develops between the bulk solvent and the stagnant film or the solvent which is in direct contact with the drug particle, which further enhances the rate of mass transfer and increases the overall extraction efficiency of the process. And same happens in the triple maceration. As the name indicates triple, it means we are dividing the total duration into 3 equal parts, and in case of the example which we discussed above in double maceration, say for example you are extracting the drug for 3 days, then after 24 hours we will change the solvent. Again after 24 hours we will change the solvent and we will mix the washing. So this is how we can increase the efficiency or overall efficiency of the maceration process by repeating the maceration. Now in some cases we can also go for the change of the nature of the solvent, or in other words, if I say, we can always go for the pre-treatment of the drug material in order to increase its extraction efficiency, and this is specific for specific drugs. Say for example, I have taken an example of preparation of opium tincture, and in this, for the first 6 hours, opium slices are treated with the boiling water, and this treatment helps in loosening up of those slices. They help in disintegration of those slices, they help in swelling up of those slices when we treat them with the boiling water. And once the drug is swollen or disintegrated, it helps in the formation of microcapillaries between those drug particles, which will help in entry of the solvents which makes the entry of the solvent quite easy into the drug matrix. Now this treatment, we consider it as a first maceration. So once the first maceration for 6 hours is complete, then we change the nature of the solvent. In place of water, now we use hydroalcoholic solvent, which is normally 90 to 80% of aqueous ethanol. Now since treatment of water has caused the swelling of the drug, it has causes the formation of capillaries between the drug particles. Now this aqueous ethanol will easily enter into those drug particles and will extract out the desired material, and the resultant extract or tincture is less gummy in nature. So this is how you can always modify the extraction procedure depending upon the type of the drug you are dealing with. Another type of maceration is the hydrolytic maceration, wherein we expose the drug towards the hydrolysis of certain constituents which are required. So for this hydrolysis, we treat that drug material with specified enzymes depending upon the nature of their cellular wall or the cellular structure. They can be proteases, they can be cellulosases, they can be pectinases, so which break down the cell wall, which further causes the hydrolysis of the constituents that are present in the plant matrix. Either they are present in the glycosidic form or in any other form, we can hydrolyze them and then we extract it. So I have taken example of the preparation of wine. We know that wine is normally prepared with grapes, and grapes contain various sugar bound glycosides. So what we do is we add cell wall degrading enzymes to the mixture, which causes the breakdown of the cell wall, and eventually crushing is another step that is involved in the preparation of the wine. This breakdown of the cell wall followed by the crushing helps in the proper extraction of the constituent that are present in the grape cell walls or in the cells of the grape tissue. This hydrolysis, this type of hydrolysis has been found to increase the organoleptic characters of the wine, such as aroma, taste, color, or the flavor. And it has been found by using this hydrolytic maceration process, the overall quality of the wine was significantly improved in comparison when it was prepared by using a normal maceration process. Now we move on to the modification for the large scale maceration process, because in industry we are not dealing with 1 or 2 kg of drug. We have to extract the drug in terms of hundreds of kgs at the large or the industrial level, and this is the major problem when it comes with the shifting of the process from the laboratory scale to the industrial scale. One of the major problem is the shaking or the agitation that we provide at the laboratory scale, and when we have to shift that method up to the industrial level. Then further, whenever we are dealing with an extraction process in an industrial scenario, then the process should be economical and it should provide the better yield when we are talking about the extraction. The overall cost to benefit ratio should be always high. The benefit should always be towards on the higher side with respect to the cost. So economical, and it should have a better yield. So keeping these points in mind, certain modification of the maceration process has been done for the industrial level, and the first modification is the circular extractions. Now as I told you that tons of material is being extracted in industry in comparison to 1 or 2 kg of the drug material at the laboratory level. So if we require say 20 liters of the solvent to extract out 1 kg of drug, we may require 100 liters of the solvent to extract the tons of the drug material. So the cost level of the solvent will increase tremendously. So in order to decrease that cost in the circulatory extraction, we recycle the solvent with the help of the pump. So as you can see in the diagram, there is an extraction chamber, cylindrical extraction chamber having plant drug material, and at the bottom of that cylindrical chamber there is a pipe which is attached with the pump. So once we spray the solvent onto the drug material, allow the drug material to stand for specified period of time, and once this time is over, we open the valve of the pipe which is running from the bottom of the extracting chamber, and this pipe, with the help of pump, we recirculate the solvent. So once the extraction is complete, we remove the drug material, we add the fresh solvent, we add the fresh drug material, and this solvent is circulated again with the help of pump. So this is how we can reduce the overall cost of the solvent when we are dealing with large amount of the drug material to be extracted with the help of the maceration process. Another modification is the multi-stage extraction procedure. Now if we talk about the circulatory method, because we are circulating the same solvent once or twice to extract the drug material, this causes the problem of formation of equilibrium between the solvent and the drug particle, and eventually the mass transfer rate decreases considerably, thereby affecting the overall extraction procedure. Now to overcome that problem of equilibration in the circulatory extraction method, multi-stage extraction has been designed. Basic concept is same, but we have added 3 additional receiver container 1, 2 and 3, as you can see on in the picture. Again, the drug material is packed in the cylindrical extractor. It is sprayed with the solvent. Now this solvent, which is after specified period of time, this solvent is then shift on to the container number 1. Container 2 and 3 are also filled with the solvent. Once this solvent enters into container 1, the solvent from container 2 is then again sprayed over to the solvent. Likewise, this series of washing of the drug with the solvent or the maceration of drug with the solvent continues, and the solvent is circulated. Once the solvent that is present, fresh solvent that is present in the container number 3 is used for the extraction, by this time the drug has already been extracted with the help of the solvent that was present in container 1 and 2, and this extract that is obtained after treating the drug material with the solvent in container number 3 will be the least concentrated. So once this extraction with the solvent in container 3 is completed, this solvent is then entered into container number 1. This is how this cycle keeps on rotating, and we get a concentrated extract without attaining the equilibrium, and this helps in increasing the overall mass transfer rate. The major advantages of the multiple extraction method is we can increase the number of extraction just by adding the receiver containers. In the previous picture, we have seen the 3 solvent containers were there, container 1, 2, 3. If you want to further increase, we just have to add another solvent container, that is container number 4. So you can increase the number of the extractions by just adding the additional solvent container in the whole setup. Now, if more extraction stages are required, it is only necessary to have more receivers. This is what we were talking about. The last treatment of the drug before it is discharged is with the fresh solvent. So as we discussed, that whenever the drug is treated with the fresh solvent that is present in the third container, the drug has already been extracted with the solvent in the first 2 containers. So that fresh solvent from the third container will cause an overall washing of the drug, and whatever remnant amount of the phytoconstituents, if they are left in the plant matrix, they are washed out, they are extracted with the help of fresh solvent, and this process or this step increases the overall yield of the extraction process. So these are few of the major advantages of the multiple maceration extraction procedure. So this multiple stage extraction has further been modified by using a extraction battery. Here you can, in this picture you can see there are 3 containers. I have taken an example of the simplest form of the extraction battery. Now the major advantage of extraction battery is all these 3 containers are attached to each other with the help of pipings, and you can remove the component of any of the container at any given time period. Once the cycle is complete, you can remove the contents of the container A. You can also remove the contents of container C or B, depending upon the type of the cycle going on into the extraction procedure. Now, say for example, these 3 containers, container A, B, and C, they are filled with the drug material, and we have added fresh material to the container number C. Now, whatever solvent was present in the container A, in the container B, is now shifted onto the container C, which contains the fresh drug material. And the solvent that was present in container A then will be shifted to container B. And to the container A, we will add fresh solvent. Now, fresh solvent is added when the drug material has already been extracted with the 2 maceration processes or by the 2 solvents. So we add fresh solvent to the container A, which contains partially exhausted drug. Now in the second step, what we will do is we will remove the solution from the C, because the C has charged with a fresh drug and it was extracted with the solvent from the container B. So the extract that will be present in the container C will be highly concentrated. So we will remove that from the container C and put it under the concentration process. Then we will transfer the solvent from B to C and transfer from the solvent from the container A to container B. Mind you, the drug material present in the container A has already been extracted by 2 maceration processes. So the drug present in A is now completely exhausted, and we will remove the drug material from the container A. Then we will charge the container A with the fresh drug. Now whatever solvent that was added to the container C after removing the initial solvent will cycle back to the container A, and from container B to container C, and from container A again to container B, or you can always add a fresh solvent to the container B. So this is how this cycle keep on we continue this extraction cycle till the whole batch of the drug is completely extracted. Now there are few advantages of this extraction battery. We are recycling the solvent to cut down the solvent cost, 1. Second, we are simultaneously extracting the drug material in 3 different containers with the same solvent. So we are able to extract out large amount of drug in a single cycle, and this helps in reducing the overall cost of the solvent, it helps in reducing the overall energy cost which is used to concentrate the extract. It helps in reducing the overall labor cost because we are extracting out the large amount of a drug in a single cycle. Now we move on to another type of maceration process which is known as digestion. Now this is nothing but this is maceration at an elevated temperature, and this temperature is normally kept below the boiling point of the solvent that is being used for the extraction. So slightly elevated, you can also consider this elevation above the room temperature. And during this process also you can provide certain agitation or shaking to the container to increase the overall extraction efficiency. Now what happens with this increased temperature? The viscosity of the solvent decreases and the penetration power of the solvent into the drug matrix increases. Further, the increased temperature also causes the solubilization of the constituent into the solvent. This overall increases the extraction efficiency. And since solubilization power increases, penetration of the solvent increases, the overall time taken to extract the drug reduces. Vis-a-vis, it also decreases the volume of the solvent that is being involved in the extraction of the drug. So friends, this was about the maceration and the digestion process. In the next lecture, we will discuss about another conventional technique, that is infusion. Thank you.